Now, before we start talking about how light energy is absorbed by the pigments in plant cells inside the chloroplasts in order to make glucose, it's important to be able to write the chemical equation for cellular respiration, because we know all the cells of a plant respire, they require glucose and they break that down inside their cells. And the chemical equation for photosynthesis, because the end product of photosynthesis, as we know, is glucose. So if you want to pause the video, you can write down these two equations, and then very shortly I'll show them on this slide. Okay, so the first equation for cellular aerobic respiration. Well, we know glucose is a monosaccharide. We know this is the starting molecule that gets broken down in the cytoplasm of all plant cells. Now, we also know that oxygen is the final electron acceptor at the end of the ETC inside the mitochondrion in the inner mitochondrial membrane. Now, the oxygen is very electronegative. It pulls electrons through the ETC. So the low energy electrons, as they exit the ETC, get combined with oxygen and also some protons to form water. So the oxygen combines with the electrons and some protons to form molecular water, and that's a waste product of respiration. We know carbon dioxide is produced as a waste product, and in aerobic respiration this comes from the link reaction and also the Krebs cycle. So for each glucose molecule we get 6 CO2 being excreted from the cell as a waste product. Now, of the chemical energy in the glucose, we know that ends up as ATP. Now, 34 of these ATP are produced by what we call oxidative phosphorylation at the electron transport chain, and 4 of the ATP are produced by substrate level phosphorylation, and we get that from glycolysis, where we get 2 ATP net, and we also get 2 ATP from the Krebs cycle, which goes around twice per glucose. Now, if we look at the equation for photosynthesis then, well, we know the carbon dioxide produced in cells like the spongy mesophyll and the palisade mesophyll is quite often the substrate that gets used and fixed in order to make glucose. Now, other carbon dioxide molecules come from the atmosphere through gas exchange. Now, we know stomatal pores, when they're open, they allow CO2 gas to come into the air chambers inside the leaf. We know there's a thin film of water around both spongy and palisade mesophyll, which dissolves the CO2 so it can actually diffuse into these cells, and inside the cell, in the cytoplasm, that CO2 will then diffuse into the chloroplasts, and it's inside the chloroplast that the CO2 becomes fixed with, as we'll see a bit later, a molecule called ribulose bisphosphate to form the glucose. So glucose is an end product of photosynthesis that is then respired by all the cells of the plant. So we know plants, as we said previously, are autotrophs, and that means they can make their own glucose for respiration in all their cells. Now, oxygen is a waste product of photosynthesis. So some of the oxygen released will actually be used in respiration, and some of the oxygen will be released through gas exchange and then out of the stomatal pores. Now, when we talk about the absorption of photons of light, we need to understand there's different wavelengths of light that correspond to the different colors that we sometimes can see. So if we have a look at the electromagnetic spectrum, which is this grey bar here, we can see there is light of different wavelengths, and each wavelength of light will have different energies associated with it. So in terms of visible light, that's a very small part of the electromagnetic spectrum, and it's this bit right in the middle. Now, we know visible light is split into different colors if it's passed through a prism, and these will be from dark blue and violet on the left hand side through green, yellow, orange and red on the right hand side of the visible spectrum. Now, the violet and blue light corresponds to the higher energy photons of light. So these are what we call 400 nanometers to about 450 nanometers on the left-hand side. So we call them high energy photons of light. Now, red light is slightly lower energy, so the 700 to 800 nanometer range from here to here. This is slightly lower energy photons of light. Now, we know green plants, they have chlorophyll A as one of their photosynthetic pigments, and we know that reflects green light. So the wavelengths of light in the middle of the visible spectrum, from about 500 nanometers to about 600, that is green light that predominantly gets reflected by the photosynthetic pigments. So the energy contained within wavelengths of light is related to the wave. So if you have a high energy wavelength of light, it might look something like this at the top. So this might be, for example, 500 nanometer blue light. And as we see from this diagram here, it's the peak of the wavelengths, and that's the distance that we measure in nanometers. Now, if it's a lower energy photon of light, it might look something like this. So we can see the distance between the peaks of the wavelengths is a lot longer. So this might be something like 800 nanometer light, and this contains less energy. And this would be from the red part of the spectrum on the right-hand side. So if we had a look at this slide, we can see it's red in color. So if we look at the electromagnetic spectrum, we know the visible part of this spectrum is the bit in the middle. And we know the wavelengths go from 400 nanometers to 800 nanometers. So what light is being absorbed by the background on this slide? Well, we know it's not red light because that's been reflected, because that light, that energy of light, ends up hitting the retina cells at the back of your eye, which is why we perceive it as red. So the light that's being absorbed is everything excluding the red part of the visible spectrum. So from the left-hand side, all this visible light is being absorbed by this red slide. So the violet, the blue, the indigo, the green, the yellow and the orange. And it's the red light that's being reflected from the background of the slide into your eye. So there you go. 400 to 700 nanometers is being absorbed, and about 700 to 800 nanometers is being reflected. So again, we can use the yellow background and look at the different wavelengths of light. Now we know the visible spectrum is a small portion of the electromagnetic spectrum in the middle, and we know if we can see yellow colors, well, that must be being reflected by this slide. So this light in the middle of the visible spectrum is being reflected, and all the other wavelengths of light from 400 nanometers to just under 600 nanometers, and then about 610 nanometers to 800 nanometers is being absorbed by the yellow background. So violet to green lights, and then the orangey to red light is being absorbed. And we know the yellow light from 590 to 610 nanometers is actually being reflected. Now, when we talk about the green colors of the leaves of plants, we now know that when we look at the electromagnetic spectrum, we know the visible light has different wavelengths. We know the green colors must be because that wavelength of light is being reflected by these particular pigments that we'll talk about later. And all the other wavelengths of light, excluding the green, must be absorbed. So the violet, blue, indigo, yellow, orange, and red parts of the spectrum are being absorbed by photosynthetic pigments, whereas the green light is being reflected. So these are the nanometers that we could jot down. So the green part of the visible spectrum is between about 520 nanometers, so about here, and about 580. So this is the light that's being reflected by chlorophyll A, which is the primary photosynthetic pigments that we find in the photosystems in the thylakoid membrane. Now, before we start to talk about the light-dependent stage, it's important to recap the structure of a chloroplast. So we know a chloroplast is an organelle that we find in certain plant cells, in particular spongy and palisade mesophyll cells. So this structure would be about 5 micrometers in length from the left to the right-hand side. So it equates to about the size of a bacterium. They're actually very similar in size. And it was once thought that the chloroplasts were unicellular organisms that did live independently as a single separate organism. Now, these possibly billions of years ago did become incorporated and engulfed by a larger prokaryotic cell. And that might have been the start of the formation of eukaryotic cells that contained organelles. And we call that endosymbiosis, because these organelles that we now know are part of the larger eukaryotic cells, they could once have been free-living, independent organisms on their own. Now, we know a chloroplast has a double membrane. So it's likely when this process did occur billions of years ago that the outer membrane of the chloroplast, and we know it's got an outer and an inner membrane, it might well have been part of the cell membrane of that cell that did first engulf these chloroplasts. Now, because they can photosynthesize inside the chloroplast, this was beneficial to the larger eukaryotic cell that might have engulfed it. So it was retained by that cell and became incorporated and part of that cell, which then got replicated. And we now know plant cells, all of them or most of them, contain chloroplasts that can photosynthesize as part of the cell itself. So if we look inside the chloroplast, the fluid organic material, this kind of light pink color, is the stroma. And this is the site where the glucose is actually synthesized. Now, the stroma surrounds these green discs that we call thylakoid discs. Now, you can see there's many stacks of thylakoid discs, and that increases the surface area dramatically. There's a very large surface area inside the chloroplast. Now, they're green in color, the thylakoid discs, because in the membrane of the thylakoid disc we find the photosynthetic pigments. And these pigments fall into different types. The primary pigments are those like chlorophyll A, and the accessory pigments are those such as the carotenoids like beta-carotene and xanthophyll, as well as chlorophyll B. So the accessory pigments are also in the thylakoid membranes, and they will absorb certain wavelengths of light, and they will pass that excitation energy down to chlorophyll A, and that will then be utilized in the light-dependent stage. Now, we know a chloroplast, you can see these red dots inside the stroma. So I'll just highlight them in yellow. So we can see the yellow dots here. Sorry, the red dots. Now, these represent 70S ribosomes. So we know the ribosome is the site for translation of messenger RNA into a protein. So the chloroplast can make all its own proteins, and these might be proton pumps, they might be enzymes involved in the Calvin cycle to make the glucose in the stroma. So if a chloroplast has its own 70S ribosomes to make proteins by translation, where does the mRNA come from? Well, we can see there is small circular DNA inside a chloroplast. So all the genes, or alleles we could say, which are variants of a gene, all the alleles that code for the mRNA, that codes for the proteins inside the chloroplast, are located on this small circular DNA inside the stroma. So anytime we've got genetic material, we will get messenger RNA, and that is then translated at the ribosomes. Now we can see the dark black structures inside the stroma. They are starch grains. So once glucose has been made inside the stroma by the Calvin cycle, a lot of that glucose, because it's not used straight away by the plant cell, is stored as starch. Now we know starch grains are made up of two types of polysaccharide, amylose and amylopectin. Now amylose has thousands of alpha glucose monomers linked together by alpha 1 to 4 glycosidic bonds, and that's in a spiral shape. Now amylopectin has both alpha 1 to 4 and alpha 1 to 6 glycosidic bonds. Now the alpha 1 to 6 glycosidic bonds are the branch points, because we know amylopectin is a branched polysaccharide, and that's because glucose can then be released simultaneously from the ends of the branches to be used inside the plant cell for respiration to release the energy as ATP. Now we're going to see shortly that the thylakoid disc, inside the thylakoid disc we get protons being pumped in here. So inside the thylakoid discs is going to be very acidic, a very low acidic pH. And that's going to build what we call the electrochemical gradient that drives the synthesis of ATP that's needed to synthesize glucose in the stroma. Now, if we look at the absorption spectrum, this is a graph that shows us the quantity or the amount of light energy that's absorbed by each of the photosynthetic pigments at the different wavelengths of light. So all you have to do when you get a graph, really, to start with anyway, is to look at the labels on the x-axis along the bottom and the y-axis going up the left. Now we know the x-axis represents the independent variable, which in this case is the wavelength of light. And the y-axis on the left represents the dependent variable. And that is the factor that gets measured. So we know the relative absorption on the y-axis represents how much light energy is being absorbed at different wavelengths of light for each of the pigments. So what we can see is, if we start with chlorophyll A, which is actually the purple line, the peaks for chlorophyll A occur at about 420 nanometers. So that's the highest peak on the left, and that represents the blue light. And the second peak for chlorophyll A is on the right hand side, and that occurs at about 630 nanometers, and that represents red light. So we know the highest quantities of light being absorbed by chlorophyll A are the blue part of the spectrum and the red part of the spectrum, because that is where the highest levels of light are being absorbed on the y-axis. Now for chlorophyll B, we can see there's a peak at about 450 nanometers. So that represents where there's a greater amount of light energy being absorbed by this pigment over all the different wavelengths. It's about 450 nanometers. So that represents obviously blue lights, the blue end of the spectrum again. So we know chlorophyll A is the primary pigment. It absorbs a large amount of light energy of both the red on the right hand side and the blue on the left hand side part of the spectrum. We know chlorophyll B is an accessory pigment, and that absorbs predominantly about 450 nanometers in the blue part of the spectrum on the left. We know the carotenoids include beta carotene and xanthophyll, and these are accessory pigments that absorb different wavelengths of light compared to both chlorophyll B and chlorophyll A. And they have the highest amount of light energy being absorbed at about 475 nanometers across the visible spectrum. So the absorption spectrum is defined as the quantity of light energy being absorbed by each photosynthetic pigment across the spectrum of visible light wavelengths. Now we've got the absorption spectrum at the top, so we know it's the quantity of light being absorbed by the different pigments at all the different wavelengths of light. And we said earlier that chlorophyll A has the most light energy being absorbed in the blue part of the spectrum and also at the red end of the spectrum, There's lots of light energy, lots of photons of light, of blue light and red light being absorbed by chlorophyll A here. But what does this not tell us? Well, it does tell us the quantity of light being absorbed by each pigment at all the different wavelengths, but it does not tell us what this light energy is actually being used for. Because remember, the dependent variable, the y-axis going up the left-hand side, is the factor that gets measured. So this actually does not tell us anything about photosynthesis itself. So in order to correlate this graph with photosynthesis, we need to look at the rate of photosynthesis at the different wavelengths of light. Now what we can see is at the blue end of the spectrum on the left hand side, so about 400 nanometers to about 450 nanometers, we've actually got a very high rate of photosynthesis. And also on the right hand part of the visible spectrum from 600 to 700 nanometers, we have again a high rate of photosynthesis. So we can correlate the two graphs. The absorption spectrum shows us the amount of light energy being absorbed in the blue and the red parts of the spectrum. And we can now say with a little bit of certainty that this light energy is probably being used directly in photosynthesis, because the rate of photosynthesis at these parts of the visible spectrum are very high, where we've got absorption of large amounts of blue and red light. Now, one of the first experiments to demonstrate that red and blue light was being absorbed by the photosynthetic pigments and used in photosynthesis was by a scientist called Engelmann in 1887. And his experiment involved a eukaryotic unicellular organism, Protoctista, called Spirogyra. Now, the chloroplast, the single chloroplast inside a single Spirogyra cell, is actually a ribbon shape, and it's in a helical shape. So it doesn't look like the typical chloroplast you find in a plant cell, but it does the same sort of job. Now, what he did, he shone white light through a prism, and that splits white light into all the different visible wavelengths that we see, from violet, blue, indigo, green, yellow, orange, and red on the right hand side. And he placed Spirogyra in a container. Now, next to this container, he had a membrane, so a synthetic membrane, and he then had aerobic bacteria that we call obligate aerobes, because they require oxygen in order to divide, and they respire aerobically. So what he did was to look at the accumulation of these obligate aerobe bacteria around the Spirogyra according to which wavelengths of light were being absorbed by that part of the chloroplast inside the Spirogyra. So if we look at the picture in a bit more detail, we can see the blue end of the spectrum on the left hand side. Just change that to blue. So the blue light on the left hand side, that's been absorbed by the Spirogyra. Now we can see there's an accumulation of these obligate aerobe bacteria in the medium around this part of the chloroplast. Now that's because high levels of oxygen are being produced, because we know from photosynthesis one of the waste products is molecular oxygen, and that comes from, as we'll see in a minute, photolysis. So one part of photosynthesis is to split water into hydrogen ions, electrons, and oxygen, and we know that's given off as a waste gas. So these aerobic bacteria could utilize the oxygen, and they accumulated in high numbers around part of the Spirogyra that was absorbing the the blue light. Now we know the red light on the right hand side, again we can see an accumulation of these obligate aerobe bacteria around the Spirogyra that has the absorption of red light in its chloroplasts. So we know oxygen is being released from the Spirogyra in this area, and that concentrates these bacteria because they can utilize the oxygen for aerobic respiration. Now we see in the middle where the green light has been shone on the spirogyra, there's very little accumulation of these bacteria, and that's because the green light is not being absorbed by the photosynthetic pigments, it's being reflected. So this light energy is not being utilized in photosynthesis, and so there's no production of oxygen at these portions of the spirogyra, which means there's a lack of accumulation of the obligate aerobe bacteria. Now, another experiment that you could be asked about is the use of filters. Now, we said earlier that certain colors, like if you've got a wall that's been painted a red color, that will reflect red light, like the background on the red slide reflected red light. Now, the filters work in the opposite way. In a filter, if it's a red filter, it will only allow red light through it, such as a blue filter will only allow blue light through. So we can start to investigate the rate of photosynthesis at different wavelengths of light, depending on the type of filter that we've used. So you might have this set up in the picture where we've got an underwater plant that can photosynthesize. So that might be pondweed, for example. And we know that's going to take in carbon dioxide and it's going to make glucose in its chloroplasts, in its stroma, and it's going to give off oxygen gas. So one way would be to take the red filter and wrap that round this apparatus, so when light is shone through the red filter, only the red light can go through the filter and be absorbed by the photosynthetic pigments of the pondweed. Kabomba is another good plant material to use for this experiment. Now, in the extension activity on the right hand side, it does say, what is the role of hydrogen carbonate ions dissolved in the water? Well, we know these underwater plants require carbon dioxide, so the hydrogen carbonate ions, they actually dissolve in the water and they provide the carbon dioxide that's needed by the chloroplasts in the production of glucose. Now, one way of measuring the rate of photosynthesis would be to measure the number of oxygen bubbles being produced over unit time. So, for example, if we used a green filter, we know the green filter would only allow green light through. It would reflect all of the colors of light, including red and blue. So if you use a green filter, the number of oxygen bubbles that we would expect would be quite low because green light is reflected and not absorbed by the photosynthetic pigments. So which filter is going to produce the highest rate of photosynthesis? Well, it's likely to be the blue filter, because the blue filter placed around this apparatus will allow blue light through only. We know blue light is absorbed by chlorophyll A, chlorophyll B, the carotenoids like beta carotene and xanthophyll. So all the photosynthetic pigments absorb blue light, and that light energy is used and converted into chemical energy in the form of glucose. And the more glucose you end up with, i.e. the higher the rate of photosynthesis, the more oxygen gas will be given off. Now, the extension says, how could we make this experiment more accurate? Well, instead of counting the number of oxygen bubbles being produced over unit time, we could actually collect the oxygen gas using a gas syringe. Now, a gas syringe will collect the O2 gas being given off, and it will give us a measurement of volume. So we can actually measure the volume of oxygen gas being produced as opposed to counting the bubbles. Now, what else could we keep the same in this experiment? So in any experiment, you only change one factor. And this factor that's being changed, i.e. the independent variable, is the wavelength of light being shone onto the underwater pondweed or kabomba. Everything else is kept the same. So the type of plant will be kept the same. Actually, you would probably use the same plant itself a short time later, because that's exactly the same. The concentration of hydrogen carbonate ions has to be kept the same, because we need the same concentration of carbon dioxide, because that's used in photosynthesis. The temperature of the water will be kept exactly the same, and you might actually do that with a thermostable water bath. where you put the beaker inside the thermostable water bath. The distance the light source is from the plant is important because that is to do with the intensity of the light has to be kept the same. The pH of the solution has to be kept the same, so you could add a buffer to that to keep it a consistent pH. So these are some of the factors that we would try to keep the same. They're called control variables. So when we talk about photosynthesis, it's important to talk about the different pigments. Now, these pigments are collectively arranged in what we call a photosystem. And there's two different photosystems. We're going to start with photosystem 2 on the left-hand side. Now, photosystem 2 is made from accessory pigments that absorb a wide range of different wavelengths of light. So these circles that we can see in light blue are the accessory pigments. So things like chlorophyll B, beta carotene, and xanthophyll. And they absorb a wide range, of different wavelengths of light. So it's important to reference the wavelengths of light and not just say wavelengths on its own. So when these absorb photons of light at different wavelengths, they pass excitation energy down towards the reaction center. And that reaction center is made of two chlorophyll A molecules. Now, from these two chlorophyll A molecules, two electrons become excited to a higher energy level. And these two high energy electrons are emitted from photosystem 2, so they're released from photosystem 2, and they pass down what we call an electron transport chain. Now, the second photosystem is actually photosystem 1, and we find that on the right-hand side. Now, again, this has accessory pigments like beta carotene, xanthophyll, and chlorophyll. So we just put in chlorophyll B. And these accessory pigments, we said, absorb a wide range of different wavelengths of light. Again, the excitation energy is passed down to the reaction center. And the reaction center here is made from two chlorophyll A's. And again, two electrons become excited to a higher energy level. Now, we know chlorophyll A absorbs in the blue and the red end of the visible spectrum. So not only does it receive excitation energy from the accessory pigments, chlorophyll A, two of them in the reaction center, can also absorb red and blue light. Now, the chlorophyll A in PS2, the red end of the spectrum, it absorbs at 680 nanometers. But the chlorophyll A in PS1, over on the right hand side, that absorbs blue light but red light at 700 nanometers. So we call the reaction center of photosystem 2 P680, and we call the reaction center of photosystem 1 P700. Now, the collection of accessory pigments is called the antenna complex, because we've got so many accessory pigments that they can absorb all this light energy and pass that down as excitation energy. So it's like an antenna that amplifies the signal, and that energy gets passed and helps to excite the two electrons from chlorophyll A, the two chlorophyll A molecules in the reaction center. Now, the electrons that get released from photosystem 1, they don't get passed down the ETC normally, but what happens is they get used and emitted, and they're used to reduce the final electron acceptor, which in this case is NADP, and that becomes reduced NADP. So photosystem 1 is directly involved in producing reduced NADP. And these are the two high energy electrons that are used to do so. So this is another view of the photosystem. Now we can see here, this is the antenna complex, and that's made of the accessory pigments, beta carotene, xanthophyll, and chlorophyll B. We know the reaction center is at the bottom, and that's made of two chlorophyll A molecules, each of which will emit a high energy electron. So we get two high energy electrons being released from this photosystem. And we find these in the thylakoid membrane. So we're going to talk about a process called non-cyclic photo-phosphorylation. Now in this picture we can see a phospholipid bilayer, and that's the membrane of the thylakoid disc. So the polar head groups are in orange, and we can see the fatty acid tails, the hydrophobic fatty acid tails as well. So that's the thylakoid disc membrane. Now the space outside is the stroma, and the space inside at the bottom of the slide, well, this is the space inside the thylakoid disc, otherwise known as the thylakoid lumen. Now we can see both photosystems on this slide. On the left is photosystem 2, and on the right is photosystem 1. Now the darker green circles, these represent the accessory pigments. So things like beta-carotene, xanthophyll, and chlorophyll B. And we find these in the antenna complex of both PS1 on the right and PS2 on the left. Now, before we even talk about the absorption of light energy, we're going to talk about one of the substrates, or one of the starting materials needed for photosynthesis. Well, we know it's water. Now, the higher the rate of photosynthesis, the higher the use of water we're going to see. So there's an enzyme attached to photosystem 2 on the left-hand side, and this is activated by light energy. And what this enzyme does, it splits water in a process called photolysis. So water is split into electrons, protons, and oxygen gas. Now this is the oxygen gas that is given off as a waste product of respiration. So the rate of photosynthesis, we could look at how much water is actually being utilized, but also how much oxygen gas is being produced, as well as how much carbon dioxide will be used later and how much glucose will be synthesized. Now we know the accessory pigments absorb different wavelengths of light, and they pass excitation energy down to the two chlorophyll A molecules at the reaction center of photosystem 2 on the left hand side. The two electrons released from the two chlorophyll A molecules become excited to a higher energy level, and these high energy electrons, they get emitted from photosystem 2. That means they get released from photosystem 2. And what happens is a primary electron acceptor within the thylakoid membrane binds these two high energy electrons, and it passes them through a proton pump and then through a second electron carrier to the reaction center of photosystem 1. Now as the high energy electrons pass down this electron transport chain made of the two electron carriers and the proton pump, the electrons lose energy, and that energy is utilized by the proton pump to allow it to change shape. And that proton pump is then going to pump, by active transport, protons from the stroma into the thylakoid disc. So inside the thylakoid lumen, or the thylakoid disc, we get an accumulation of protons. So the pH drops, becomes very acidic and very low because of the accumulation of hydrogen ions. Now this is going to be what we call an electrochemical gradient. And we see an electrochemical gradient in respiration as well. And that's built up in the intermembrane space between the outer and the inner mitochondrial membrane. Well, here in photosynthesis, the light-dependent stage, the electrochemical gradient is built up inside the thylakoid disc. So the whole point of having two electrons energized to a higher energy level is that as they pass down the electron transport chain, they lose energy, and that energy is harnessed by the proton pump to build the electrochemical gradient. Now, what is one other role of the splitting of water? It's not just to produce oxygen gas. The main role is that the electrons released from photolysis replace the low energy electrons that were lost from chlorophyll A. So photolysis is really important to help replace and replenish the electrons lost from chlorophyll A, because obviously the low energy electrons in chlorophyll A became excited to a higher energy level when light energy was being absorbed, and they were emitted from that photosystem. Now we know light also hits the accessory pigments and is absorbed by these accessory pigments in the antenna complex of photosystem 1 on the right hand side. Excitation energy is passed down to the reaction center in PS1. 2 electrons become excited to a higher energy level from the 2 chlorophyll A molecules in that reaction center. Now as these electrons pass to a higher energy level, again, these are going to be emitted from photosystem 1, very much like photosystem 2. And the electrons that come from the ETC, the low energy electrons, at this electron carrier here, they're now used to replace the low energy electrons that were originally in the chlorophyll A of photosystem 1. So we know there's 2 times chlorophyll A in the reaction center of PS1. Now, the 2 high energy electrons being emitted or released from photosystem 1, they don't pass down the ETC this time, but instead in non-cyclic photo-phosphorylation, they actually pass to a second electron acceptor and then to another enzyme that is going to combine the 2 electrons with NADP with also some hydrogen ions to form reduced NADP. So the protons that have been used to build this big electrochemical gradient inside the lumen of the thylakoid disc, they now move down through the stalk particle through a proton channel. That potential energy is utilized by the ATP synthase enzyme to synthesize ATP from ADP and inorganic phosphate. So the energy, the potential energy in the electrochemical gradient has been used to build ATP, which contains that chemical energy. The protons move back into the stroma, and those protons combine with the electrons that are emitted from photosystem 1 to form reduced NADP. So as the electrons move down the electron transport chain, we know they lose energy like this. The electrons are used to replace those that become excited to a higher energy level in PS1. The high energy electrons from PS1 get emitted and get used by an enzyme to combine them with protons and NADP to form reduced NADP. So the whole point of non-cyclic photo-phosphorylation is the production of 2 molecules, ATP and reduced NADP. And both ATP and reduced NADP will both be needed in the stroma in the light-independent reactions to make glucose from carbon dioxide. Now, the energy of the electrons can be represented in what we call the Z scheme, and this represents the energy from the 2 electrons in chlorophyll A in photosystem 2 on the left hand side. We know they go to a higher energy level because of the absorption of light energy by chlorophyll A and the passing of excitation energy from the accessory pigments down to chlorophyll A. We know they're accepted by the primary electron acceptor as they get emitted or released from photosystem 2. So these are very high energy. So we could put high energy electrons, and there's 2 of them. Now we know the high energy electrons pass down the ETC, the chain of electron carriers that includes the proton pump, which gets activated. That builds the electrochemical gradient, which is then used to synthesize ATP by ATP synthase, which is the catalytic enzyme as part of the stalk particle. The electrons lose energy as they pass down the ETC, and these 2 electrons are needed to replace the 2 electrons excited to a higher energy level from photosystem 1 on the right hand side. So we know these 2 electrons become excited to a higher energy level because of the absorption of light energy by the accessory pigments and also chlorophyll A. The 2 electrons reach that nice high energy level. They're released from the photosystem 1. They get picked up by the second electron acceptor and passed to the enzyme that combines the 2 high energy electrons with oxidized NADP, as well as some of the protons, and we're going to end up with reduced NADP. Now we know the electrons from the splitting or photolysis of water at the bottom here, so we know this is called photolysis. That's light activated. These electrons replace those lost from the reaction center of photosystem 2. So we can see at the bottom left, these low energy electrons from the photolysis of water replace those lost originally from the chlorophyll A of the reaction center of PS2. Now on the right hand side, we know the 2 high energy electrons emitted from photosystem 1 are used to reduce NADP. So we end up with reduced NADP that goes into the Calvin cycle. We know ATP is also going to be needed in the Calvin cycle, and these 2 molecules are actually involved in conversion of a molecule called glycerate 3-phosphate that you can see in the other video into triose phosphate, and the triose phosphate is then used to make glucose and also to regenerate one of the starting compounds called ribulose bisphosphate. So as well as non-cyclic photo-phosphorylation, there is a second process that we can talk about called cyclic photo-phosphorylation. So this is slightly different from what we've seen previous, because in cyclic photo-phosphorylation, photosystem 2 on the left hand side here, this is not used in cyclic photophosphorylation. Now, because the enzyme involved in photolysis is associated with PS2 on the left, there's actually no photolysis in cyclic photophosphorylation. So there's no production of oxygen in cyclic photophosphorylation, because that splitting of water does not occur in this process. Now, cyclic photophosphorylation is only going to involve photosystem 1 on the right hand side. So what we see is, we know light energy, different wavelengths of light is absorbed by the accessory pigments. Activation energy is passed down to the 2 chlorophyll A molecules in the reaction center of PS1. From these 2 chlorophyll A's, the 2 electrons are excited to a higher energy level, and they're going to get released from photosystem 1. So they get emitted from this photosystem, they get picked up by the second electron acceptor in yellow here, but this time the electrons are not passed to the enzyme that will reduce NADP. Instead, this second electron acceptor is a mobile carrier, and it can actually move within the phospholipid bilayer. So it's a mobile electron carrier, and it delivers the 2 high energy electrons to the electron transport chain. So these 2 high energy electrons pass down the proton pump through the ETC, and they lose energy as they do so. Now this activates the proton pump, and it's going to pump, as we'll see in a minute, protons from the stroma into the thylakoid disc. Now the 2 electrons, they're now low energy electrons because they've lost their energy that's activated the proton pump. So we know 2 electrons have been released from chlorophyll A in the reaction center. So we know there's 2 times chlorophyll A down here. So these low energy electrons from the ETC are needed to replace those lost from the chlorophyll A molecules in the reaction center. So you can start to see this is a cyclical process, and it keeps repeating itself. So once the two electrons, the low energy electrons, have replaced those from chlorophyll A, light energy is once again absorbed by chlorophyll A, as well as the accessory pigments that pass on excitation. The two electrons become raised to a higher energy level, and they get picked up by the second electron acceptor. That's mobile. It carries the electrons to the start of the ETC. They pass through the proton pump, and they go back to the reaction center. Now we know because the proton pump is activated, it allows the pumping of protons against the gradient into the thylakoid disc, and that builds up a proton motive force, i.e. an electrochemical gradient. So in the stroma there's a relatively low concentration of hydrogen ions, and in the thylakoid disc at the bottom there's a high concentration of hydrogen ions. So the pH is going to be a low acidic pH inside the thylakoid disc still. Now those protons can move down through the stalk particle, down their concentration gradient, back into the stroma. Now we know there's a proton channel because hydrogen ions cannot simply diffuse through the phospholipid bilayer because of the hydrophobic fatty acid tails. So the only way they can get back across the membrane is through the proton channel of the stalk particle. Now all that potential energy is converted back into chemical energy in the form of ATP. So it activates the ATP synthase catalytic domain. That allows the addition of inorganic phosphate to ADP, which is adenosine diphosphate, and we end up with ATP, adenosine triphosphate. Now, so we can start to look at some of the graphs involved in photosynthesis. Now this graph represents what's called the compensation point. Now if we look at the y-axis going up the left-hand side, we can see that represents the amount or the net uptake of carbon dioxide. So we know respiration releases carbon dioxide as a waste product, but we know photosynthesis in the Calvin cycle takes up and uses carbon dioxide. It fixes carbon dioxide to make essentially glucose. So the 0 point in the middle represents no net uptake of carbon dioxide. So if we go above this 0 point, it represents the taking in or the use of carbon dioxide. So more must be being taken in than is being lost through respiration. Now anything on the y-axis below the 0 point represents the amount of carbon dioxide being released. So that's going to be more being released as a consequence of respiration compared to the carbon dioxide being taken up to make glucose in photosynthesis. So above the 0 point, there's a net intake of carbon dioxide. So more is being taken in through gas exchange and is being fixed in photosynthesis than is being lost through respiration. Below the 0 point, there's more carbon dioxide being given out through respiration compared to the amount being taken in and used by photosynthesis. Now on the x-axis, this represents light intensity across the bottom of this graph, so increasing light intensity. Now if we look at the level of CO2 uptake in different intensities of light, what we can see is on the left hand side, the red line represents the net uptake of CO2 in low light intensity. So this kind of portion here represents a low light intensity and the net uptake of carbon dioxide. So if we got low light intensity, we know the rate of photosynthesis is going to be limited because it depends on the absorption of light energy. So low light energy or low light intensity means a low rate of photosynthesis. So the rate of respiration at low light intensity is greater than the rate of photosynthesis. And if respiration rates are greater than photosynthetic rates, the amount of carbon dioxide is going to be bigger being released from respiration compared to the amount being taken in by photosynthesis. So this portion of the graph, if we think about it, it could actually represent wilting or damage or lack of growth or even death of a plant, because the rate of respiration is a lot higher than the rate of photosynthesis. So we know glucose is used up in respiration and it produces CO2, but we know the glucose comes from photosynthesis that takes in carbon in the form of carbon dioxide. Now, what we'll see is when there's zero net uptake of carbon dioxide, we're going to call that the compensation point. So this part here. And this represents when the rate of respiration, so the production of carbon dioxide, is equal to the rate of photosynthesis, or the use or the fixation of carbon dioxide. So the CO2 being produced from respiration is equal to the CO2 being used up or fixed in photosynthesis, or the Calvin cycle. So we call that the compensation point. Now we know at high light intensity the rate of photosynthesis is going to be greater than the rate of respiration. So the rate of carbon dioxide uptake and fixation in photosynthesis becomes greater than the amount of carbon dioxide being released through respiration. So this actually might represent, if we fill this in here, this kind of shaded area under this part of the graph represents growth of the plant, because there's more glucose being produced from photosynthesis. The intake of CO2 is greater than the amount of CO2 being released through respiration. So more glucose is being produced than is being used up. So more glucose is being used because more CO2 is being fixed in the Calvin cycle compared to the rate of respiration. So we can put more glucose is greater than respiration rate, or the use of glucose. Now we know at very high light intensities the amount of carbon dioxide uptake is going to plateau somewhat, because we know stomatal pores tend to close at very high light intensity to prevent too much of the water vapor being evaporated out of the leaf, out of the stomatal pores, because that would cause wilting of the plant. So we can also look at the effect of temperature on the rate of photosynthesis. So on the y-axis up the left we have the rate of photosynthesis, and that's looking at how much carbon dioxide is being incorporated or fixed per centimeter squared per hour, compared to the temperature, which is on the x-axis across the bottom. So the independent variable is the temperature, and several different temperatures would have been used, and at each different temperature the rate of photosynthesis will have been measured as the dependent variable, and that's what's been plotted on this graph. So we can see going from 5 degrees C up to about 25 degrees C, we get an increase in the rate of photosynthesis. Now we know anytime we get a question on enzymes and temperature, we need to reference kinetic energy. So at lower temperatures there's less kinetic energy, and that means the substrate molecules and the enzymes themselves move more slowly. So if there's a slower movement of both substrate and enzyme, there's going to be less successful collisions between the substrate and the active sites of the enzymes, and therefore there's less enzyme substrate complexes being formed, and therefore less product being released. So the rate of photosynthesis is going to be lower at the lower temperatures because the substrate and the enzymes both have less kinetic energy, they move more slowly, there's less successful collisions and therefore less enzyme substrate complexes. And we know that photosynthesis involves lots of different enzyme steps and different proteins, for example in the Calvin cycle, and therefore photosynthesis is affected fairly significantly due to changes in temperature. So the optimum temperature is likely to be around 25 degrees Celsius. And here this is a balance between the most successful collisions between the substrate and the active site to get the most enzyme substrate complexes without altering the shape of the enzyme in any way. So the tertiary globular shape of the enzyme and therefore the shape of the active site remains the same and it's not changed at the optimum temperature. So the shape of the substrate will be complementary in shape to the specific shape of the active site of the enzymes. Now above the optimum temperature, the rate of photosynthesis is lower than the optimum temperature. And this is because the higher the temperatures above the optimum temperature, the hydrogen bonds in the tertiary structure of the enzymes start to break because the enzymes vibrate more at the higher temperatures. And as the hydrogen bonds break between variable groups in the polypeptide chain, it alters the tertiary structure of the enzyme and therefore alters the specific shape of the active site. So if that active site changes shape, it's no longer complementary to the shape of the substrate, no more successful collisions occur, and therefore no more enzyme substrate complexes. Now, depending on the actual increase in temperature, for example, at this point, at this particular temperature here, say 28 degrees C, not all of the enzymes are fully denatured, so there still is some photosynthesis occurring in the Calvin cycle, for example. As the temperature gets nearer 35 degrees C, we can see many more of the enzymes have fully denatured, which is why the rate of photosynthesis is actually a lot lower at the higher temperature. Now, one common exam question is to look at the concentration of carbon dioxide. Now we know carbon dioxide is one of the substrates involved in carbon dioxide fixation by the enzyme Rubisco. So really what we're looking at here is the effect of substrate concentration on the rate of photosynthesis. So on the y-axis going up the left, we're going to measure the rate of photosynthesis like we had before. So the actual uptake of carbon dioxide per hour compared to the carbon dioxide concentration on the x-axis along the bottom. So the independent variable here is the concentration of CO2 and the dependent variable is the rate of photosynthesis. Now we know atmospheric carbon dioxide is about 0.035% in the atmosphere. So some experiments we will see conducted at 1% or above, which will be a very high CO2 concentration. So that might represent this end of the data. Other experiments we see conducted at very low CO2 concentrations, so sometimes 0.003%, which would represent the data at this part of the plots that we can see. Now, under very low concentrations of carbon dioxide, we know that it's going to be limiting. So the rate of photosynthesis becomes limited by the very little concentrations of carbon dioxide present, because we know carbon dioxide is needed to be fixed with ribulose bisphosphate by Rubisco to form the unstable 6-carbon compound to form G3P, two of them that are needed to make TP, that are needed to make glucose. So the rate of photosynthesis is limited by very low concentrations of carbon dioxide in the Calvin cycle. Now, converse to that, when we've got very high concentrations of carbon dioxide, what we're going to see is, even though in this case the carbon dioxide is not limiting because there's so much of it, so it's going to be used and fixed with ribulose bisphosphate by Rubisco, so that's not a problem. We see the rate of photosynthesis actually does plateau here, so it plateaus, and that's because some other factor must now be limiting. If there was no other limiting factors, then the rate of photosynthesis would continue to increase at higher CO2 concentrations, but it doesn't. It plateaus. So what else could be limiting in this scenario? Well, it could be light intensity. Now, if light intensity was limiting, it would mean that there would be less ATP and less reduced NADP from the light dependent stage from non-cyclic photo-phosphorylation. So even if there was high levels of carbon dioxide, eventually the ribulose bisphosphate could not be regenerated because G3P would not be converted to triose phosphate, which is needed to regenerate ribulose bisphosphate. So here, light could be a limiting factor at high CO2 concentrations. Now, we also know temperature could be limiting. The temperature could be too low, so the kinetic energy of the CO2 and the Rubisco enzyme could be too low, and therefore they move more slowly and there's less successful collisions. Also, we know at high CO2 concentrations, quite often the stomatal pores will close, and that limits more carbon dioxide coming in to the air chambers. Water, we know, could also be limiting because we know water is needed in photolysis to supply the electrons that replace those that are lost from chlorophyll A in photosystem 2 reaction center. So if there's not enough water, there's not going to be enough photolysis, there's not going to be enough electrons to replace those lost from chlorophyll A. So it's going to limit non-cyclic photo-phosphorylation, which is also going to have a knock-on effect in the production of ATP and reduced NADP. Now, we can also look at light intensity. So again, on the y-axis going up the left, we've got rate of photosynthesis, so CO2 uptake and incorporation into glucose. On the x-axis, we've got light intensity at different values. So on the left, we have low light intensity, and as we progress to the right-hand side, we have much higher light intensity. Now, we know with increasing light intensity, we're going to generate more ATP and more reduced NADP from the light dependent stage. So as these go up, the rate of photosynthesis also goes up because the Calvin cycle can progress at a faster rate because it requires both ATP and reduced NADP. However, when we get to very high light intensity, we can see the rate of photosynthesis plateaus even at the higher light intensity. So here it must be some other factor other than light intensity that is the limiting factor. And again, that could be things like carbon dioxide concentration might not be high enough. So it would be the carbon dioxide that could limit the reaction, but not light intensity because that's nice and high. And that might be because at very high light intensities, we know the stomatal pores do close. And again, that limits the amount of CO2 being brought into the air chambers, into the air spaces in the leaves. And therefore, it's going to limit the rate of the Calvin cycle and the production of glucose. Now, another question we might see is what could be the physical changes in a leaf at the point where the rate of photosynthesis plateaus at high light intensity? And why would that be an advantage to the plant? Well, we just mentioned that the stomatal pores do close at very high light intensity. And the reason that's important is that the water, the thin film of water that surrounds the palisade and the spongy mesophyll cells in the leaf, if the temperature is too high, because of the high light intensity, that would evaporate too much water, and there could be too much water loss that could result in wilting of the leaf. So the stomatal pores will close to prevent excessive water loss through transpiration. Now at the bottom of this slide, we've got a graph that shows the effect of light intensity on the rate of photosynthesis for two different saplings in a wood. Now, one of the species would normally grow in direct sunlight, and the other species would normally grow in shaded conditions. So suggest which species you think grows in the shaded conditions and explain your answer. So at very low light intensity, at this point on the graph, we can see the rate of photosynthesis is actually greater for species A. Now we know in the shade there's going to be a lower light intensity. So species A is likely to be the species that has grown in the shade, has evolved to grow in the shade, because at low light intensity there's a higher rate of photosynthesis compared to species B. But we know under high light intensity, species A does not have a particularly high rate of photosynthesis. It's less than species B. So species B in high light intensity has a high rate of photosynthesis, which is why you might expect species B to have evolved in brighter sunlight compared to species A. So the rate of photosynthesis is steeper at lower light intensity. So we don't really say the curve, we say the rate of photosynthesis is steeper at lower light intensity, indicating a faster rate of photosynthesis in shaded areas for species A. Now some data interpretation. So calculate the percentage increase in the rate of photosynthesis at 0.14% carbon dioxide at 30 degrees C when the light intensity is increased from 2 to 3 arbitrary units. So in the exam, we're going to use data that were given in the question and information that were given in the question. So which of these data points is going to represent 0.14% CO2, which is quite high carbon dioxide concentrations? Normally, atmospheric CO2 is 0.03%. So here it's 0.14%, which is quite high at 30 degrees C. So it must be this set of data. Now, 2 to 3 arbitrary units in light intensity, we know is going to be across the bottom on the x-axis, because that's the independent variable. So here they've done 4 separate experiments where they've had either different CO2 or different temperatures, but each experiment, the independent variable has been the light intensity. And the dependent variable, again, has been the rate of photosynthesis. So if we just use a ruler, we can draw a line straight up from number 2 on the x-axis, and where that hits our curve, we're going to draw a line going straight across to the left-hand side, and that will give us 130 units of the rate of photosynthesis. So we're going to say 130, and then we just use the units we're given, so millimeters of CO2 per centimeter squared per hour. So always put the units in. Right, 3 arbitrary units then, because we're going to go from 2 to 3 arbitrary units on the x-axis. That would be 195 millimeters of CO2 per centimeter squared per hour. So by increasing the light intensity from 2 to 3 units, we've actually increased the rate of photosynthesis from 130 to 195 millimeters of CO2 per centimeter squared per hour. Now, the question asks for percentage increase. So in order to do percentage increase, we need the starting value, which is 130. We need how much change has been measured. So 130 to 195 is 65 units, divided by the initial starting value. So here we've got 195 minus 130 is 65, divided by the starting value, which was 130, times 100 gives us 51% increase in the rate of photosynthesis from 2 to 3 units of light intensity that have been increased. Now, what would happen to a plant if there was a lack of certain mineral ions or deficiencies in these mineral ions? Now, we know if there's a lack of magnesium in the soil, magnesium is normally absorbed through the root hair cells into the root hairs before it passes, dissolved in water, across the cortex of the root into the xylem. Normally what we see is a thing called chlorosis that you can see in this leaf on the right hand side. Now we know magnesium is needed to make chlorophyll. So chlorophyll A and chlorophyll B. So if there's a lack of magnesium in the soil, there's going to be a lack of chlorophyll synthesis in the plant cells, and so a lack of the green pigment, which is why you can see more of the yellow pigment, and also obviously some of this tissue is starting to die off because of the lack of photosynthesis. Now another mineral ion that's needed to make chlorophyll is nitrate ions. Now we know nitrate ions are absorbed again in the root hair cells of the plant, and they move across the cortex through the apoplast root and the symplast root, eventually going into being pumped into the xylem vessel, and they move up the xylem dissolved in water through the transpirational pull, the cohesion tension theory. Now it's the nitrogen from nitrates that is needed in relation to chlorophyll synthesis. But the nitrogen in nitrate ions is also needed to make other nitrogen containing compounds like amino acids that are needed to make proteins. Chlorophyll we know contains nitrogen, and other nitrogen containing compounds within the cells, things like nucleic acids, so both DNA and RNA, both have nitrogenous bases, and also things like ATP, which is the nucleotide, has adenine, which is again it's a nitrogenous base. So a lack of nitrates in the soil can result in a lack of nitrogenous, organic nitrogenous compounds like amino acids, proteins, chlorophyll, nucleic acids, DNA, RNA, and also ATP. There you will get wilting because you get a lack of cell division and therefore a lack of growth. So just to summarize what we've said, we know nitrogen is taken up as nitrate ions. It moves across the root cortex and ends up in the xylem where it's dissolved in water, and it moves up the xylem to where it's required for growth, nucleic acids, proteins, chlorophyll. If there's a lack of nitrate ions in the soil, we'll see chlorosis in the leaves, a yellowing of the leaf color, promptly due to the lack of chlorophyll production. Magnesium is the ion that we find in the middle of the chlorophyll molecule. Now it's also needed, magnesium, to activate certain enzymes such as ATPase. So a lack of magnesium can have a knock-on effect in terms of the ability of these enzymes to hydrolyze ATP.